And so, without further ado, I want to welcome you to the session on the origins of life. And our first speaker is my colleague and friend, Dante Lauretta, who is a planetary scientist and Regents Professor at the University of Arizona. And he's going to tell you about his work as an asteroid hunter. And the title of his talk is Testing Theories for the Origin of Life Using Samples of Near-Earth Asteroid Bennu. Dante.
Wonderful. Thank you, Stuart. It's a great pleasure to be here. This is my first Science of Consciousness conference, and I am really impressed and have had a great time. And I want to thank everyone for the fantastic conversations, very intellectually stimulating audience. And I hope to continue that with my presentation. So my day job is as principal investigator for NASA's OSIRIS-REx asteroid sample return mission. So let me tell you a little bit about that. OSIRIS-REx, like all good NASA missions, is an awesome acronym that explains the scientific objectives of the program, and it's also entirely my fault. But when I first started working on this program back in 2004, I started to develop the main scientific themes. Why would you go to an asteroid? Why would you collect a sample? Why would you bring it back? And I wrote down the words origins, spectroscopy, resources, and security. And I'm also a big mythology fan. I heard some people talking about the Egyptian origin myths in our session last night on space, time, and consciousness. And Osiris is a myth I was very familiar with. Osiris has an interesting dual nature as the bringer of life, the god of agriculture, probably the first pharaoh who established the civilization in the Nile Delta, but later on became the god of the afterlife. And asteroids have that same kind of dual nature. For the origins investigation, we're going after the big questions. Where did we come from? Why is Earth a habitable planet? How did life arise here? For spectroscopy, spectral interpretation, that's a technique we use to try to measure the chemistry and mineralogy of distant planetary objects. So by bringing a sample back, we'll be able to ground truth that information. For resource identification, we're looking at resources in space that we could use in situ for resource exploration, like metals for structural components, fuels for rocket fuel, life support, etc. For security, we're very interested in asteroids as potential natural disasters, and we want to understand the likelihood of such a disaster and ways that we might be able to mitigate it. And we're the regolith explorer, regolith being the loose blanket of rock and gravel that's on the surface of these airless bodies, and that's what we're bringing home for study in our laboratories. So what are the near-Earth asteroids? You're probably familiar with asteroids. They're a small population of small bodies, primarily located between the orbits of Mars and Jupiter, as you can see in this diagram here. And they are the oldest geologic samples of the solar system. I'm a geochemist by training, and when you study geochemistry and the history of the Earth, you want to understand changes through time. You go to deeper and deeper rock layers, and they formed longer ago in the past, and eventually you reach the oldest rocks on Earth, which are of order 4 billion years old, and you want to know what happened before that. You can go to the moon. The moon records some older processes than that, but the very first rocks that formed in the solar system are still preserved in the asteroid belt, and they are about 4.56 billion years old. And when we're looking for origins of life, we're looking for an asteroid that has some key compounds like water and carbon in the form of organic molecules. We see that those are relatively abundant in the outer main asteroid belt. So when we were first thinking about the mission, we thought we should just go out to the asteroid belt and we should get a sample there. But that's very energetically difficult and it takes a long time. Fortunately, Jupiter is constantly perturbing the asteroids, causing them to crash into each other. Some of them tumble into the inner solar system, and they get into very Earth-like orbits. And so it's kind of like the rocks in a stream bed. You can understand what the mountain is made out of. If you just go poke around here in the arroyo outside of the resort, you can understand that there's all kinds of interesting granites and rhyolites up on Mount Lemmon. So we're lazy astronomers. We didn't want to go all the way out to the asteroid belt. We chose a near-Earth asteroid. I started working on the project in 2004, and by 2016, we had reached this point.
Status check to proceed with terminal count. Atlas systems propulsion.
Go.
Hydraulics.
Go.
Schematics.
Go.
LO2.
Go.
Vehicle System Engineer.
Go.
Anomaly Chief.
AC is go.
Range Coordinator.
Clear to proceed.
Launch Director.
LD is go, and you have permission to launch.
Proceeding with the count. Status check.
Go, Atlas. Go, Centaur.
Go OSIRIS-REx.
5, 4, 3, 2, 1. And liftoff of OSIRIS-REx. Its 7-year mission to boldly go to the asteroid Bennu and back.
So one thing that relates to this conference is that was my out-of-body experience. There's nothing quite like spending 12 years of your career and understanding that hundreds of people's jobs are on the line. If that doesn't go well, you're going to have to explain to them that they're getting a pink slip. It was amazing. I went into space that day, literally went into space. We spent a year going around the sun. We actually came back to the Earth and we snapped this image. And if you're familiar with the overview effect, I have felt that in spades here. This is a great shot because we got the Earth and the Moon in the same field of view, which you don't usually do. And it was just amazing for me to sit there and think about that this robotic explorer that we built, spent a year traveling through space, came all the way back to the Earth, and we were able to get this amazing picture of the pale blue dot, the beautiful world that we live on here. Another year went by, and then we arrived at Asteroid Bennu. You can see its orbit shown there on the left. So the Earth is in green, Mars is the white circle on the outer edge, the Sun is at the center, and Asteroid Bennu's orbit is in blue. And you can see why we call it a near-Earth asteroid. It actually has a very Earth-like orbit, and in fact, when you look at the lower diagram, which is kind of an inclined view, you can see it directly crosses the orbit of the Earth. And because of this, we call it a potentially hazardous asteroid. It may, in fact, impact the Earth, and it has the highest probability of impacting the Earth of any known object in the solar system. Don't get too worried. The impact, if it's going to occur, and the odds are slim, about 0.05%, will be on September 24th, 2182. So you can put that in your calendar, but don't rush out and buy asteroid insurance. You don't have to quite worry about it yet. And then the object there, as we saw it during our final approach, is shown rotating. It's about 500 meters in diameter. To give you a sense, when you look at the Catalinas and you see the Push Ridge off on the western edge there, they're about 500 meters tall, so it's about the size of a small mountain flying through space. The primary objective of the mission is to bring a sample back. We did an amazing array of science. We spent all of 2019 mapping the asteroid and finding a site on the surface that would meet our expectations. But the driving requirements are to bring material home to study in our laboratories. We needed to return at least 60 grams, which is about 2 ounces of material. And then we also had a backup sampling mechanism using contact pads. Simply by touching the asteroid surface, we would grab material. We ended up flying stainless steel Velcro. As you probably know, if you stick Velcro into a sand pile, it'll get all covered in dirt. That's the strategy that we use. That's NASA engineering at its finest. But most of the technology for sample collection involves the collector head there. It looks like an air filter. It is an air filter. There's no air on the asteroid, so we bring our own air in the form of gas bottles. It's kind of like a leaf blower. You just blow the gas down into the regolith, and it expands up into that filter, and the filter traps it, and then you stow it and bring it home. The other thing I like about this photo, this is a very early engineering design, and that's what we thought the asteroid surface was going to look like. That it was going to be beach-like, sandy, and we were just going to touch down on the surface, blow the gas in, collect the sample, and back away. It is a touch-and-go strategy, so we bring the spacecraft in at about 10 centimeters per second. You can see that air filter. Those discs on the outer edge are the contact pads. Even in the computer animation, we drew the nice sandy beach. We made contact, we fired the gas, it stimulated the regolith up into the filter. We back away from the surface of the asteroid, and then we do some checkouts, make sure we got the sample that we wanted, and then we'll stow it into our sample return capsule, which is like a re-entry vehicle, kind of similar to the Apollo astronauts design, although much smaller. That sample head's about 30 centimeters in diameter. We put the entire filter into the capsule, detach it from the robotic arm, close up the capsule, and then bring it home to Earth. The site that we selected, we ended up naming Nightingale. All of the features on Bennu are named after birds, most of them mythological birds, but for the sites, the potential sites, we named them after birds that were native to Egypt. This is an impact crater, so you can kind of get a sense of the bowl-shaped morphology of the surface. All of this is real data collected by the spacecraft. We had a laser altimeter to measure the roughness and topography of the surface, and then we had an amazing suite of cameras. So this is a three-dimensional digital terrain model with the imaging data overlaid on top of it. That crater is about 20 meters in diameter. When we designed and launched the spacecraft, we had a guidance system that was capable of 25-meter accuracy. But I think you can see if we were outside that crater, there's some really massive boulders that would cause a lot of problems for the spacecraft. So we had to make it smarter. Avi Loeb, during the keynote today, talked about AI astronauts. OSIRIS-REx is an AI astronaut. It had an onboard optical recognition system. We had put in an entire map. This map was on board the spacecraft's memory. It knew all of those features in 3D, and it could predict what they would look like under the specific lighting conditions, and it adjusted its trajectory to target the location on the asteroid's surface. This is what the approach maneuver looked like. So we were in orbit, and then we fired the thrusters to start dropping down towards the surface at a point we called the checkpoint. That was about 125 meters. And then we fired again for the match point because we had to match the rotational motion so that we were basically hovering over the asteroid surface. We made a brief contact, fired the gas, and then backed away. And then this is what that looked like from the spacecraft's point of view. So now we're looking down. We're coming in at 10 centimeters per second. We made contact, and we really made a mess on the asteroid's surface. That one of the biggest surprises was that it was not a hard surface. It was like dropping into a pond of water. The asteroid regolith just flowed away from us, and we sunk in about 50 centimeters, so about the length of my arm. We went down into the asteroid subsurface. We collected a lot of material. We estimated that we got about 250 grams, so 4 times the mission requirement. For a sample scientist like me with transmission electron microscopes that image things at the atomic scale, that's a lifetime supply of material for hundreds and hundreds of cosmochemists around the world. So we had some challenges. I won't go into all of that there, but we eventually got TAG-SAM into the sample return capsule, closed it up, and it is now on its way home. We left Bennu in May of 2021, and we're on interplanetary space. So now what? So I had spent at that point in 2020, I'd been working on the program for 16 years, pretty much focused on that moment of contact with the asteroid surface, successfully collecting the sample and stowing it away. And now I get to step back and say, okay, what did we promise NASA we were going to do with the sample? So I went all the way back to my concept study report, which was submitted in 2010, and I read this statement that analyses of these samples provide unprecedented knowledge about pre-solar history through the initial stages of planet formation to the origin of life. And I was like, okay. I looked at this as a figure from the proposal there, and we said pre-solar history, easy. Solar system formation, easy. Planetary formation, easy. Asteroid evolution, no problem. Asteroid spectroscopy, great. Origin of Earth's volatiles and organics, fine. Origin of life. And I was like, okay, unprecedented knowledge. I promised unprecedented knowledge about the origin of life. How am I going to deliver on that? One of the reasons I'm here today is for help in finding that answer. So if you have ideas, come and talk to me. But we have an approach, and it's a very standard approach right now, but I'm looking for something beyond that. So let's take a look at what are we looking for. We're looking for the building blocks of life. So here I have a very simple diagram of the structure of a bacterial cell. This is a prokaryotic cell, very primitive organism, we think, similar to the last universal common ancestor of all life on Earth. It doesn't have a well-defined cell structure. It has no cell nucleus. The DNA is just floating freely in the cytoplasm. It has lots of ribosomes. There's no or very few organelles like mitochondria or chloroplasts, so they're not present in these things. They have cilia, and they have other ways of mobility. If we look at the little flow chart there, you can see the composition of a common bacterial cell. 80% water. Great. I can check off if the asteroid's delivering water. We actually did detect water on the asteroid, not as liquid water or even ice, but as clay minerals that have water locked up in their crystal structure. The other 20% 20% of a bacteria we call dry solids. 90% of that 20%, so 18% of the cell, is carbon, oxygen, nitrogen, and hydrogen. And I don't think it's a coincidence that the 4 most abundant elements of a bacterial cell are also 4 of the most abundant elements in the universe, hydrogen being number 1, oxygen, carbon, and nitrogen being number 3, 4, and 5, with helium as number 2, which is a noble gas that doesn't react. The other 10% we say is inorganic. We got things like phosphorus, sulfur, sodium, calcium, magnesium, potassium, and iron. So I could say I'm just going to tell you if the asteroid brought these elements to the Earth, and therefore if I just drop these into a test tube and stir them up, I'll get life, right? No, it doesn't work that way. We will do that. We'll measure all these abundances in the asteroid sample, but that doesn't tell us anything about the origin of life. I can already tell you all these elements are on that asteroid. The key is how do they organize themselves into the first organism? So we can take another step and say, okay, what chemical components do cells have? This is a slightly different diagram where they have 70% of the bacteria as water, so it's somewhat variable, 70 to 80%. 30% now we're looking at these chemicals. And we can see we've got nucleic acids like DNA and RNA. These are the molecules that contain our genetic code and help express it. Proteins, chains of smaller molecules called amino acids that perform both catalytic and structural functions. Carbohydrates like starches and sugars that we use for energy. Lipids, which are fat molecules that make up cell membranes. And then organelles, which are specialized and they vary among the different organisms. Like I mentioned, the chloroplasts, which are used for photosynthesis, and mitochondria, which generate energy through ATP production. So I'm like, okay, we can start to look at these compounds. We can at least see, do we have the kinds of molecules that go in to build these much longer polymers? And then I started doing a survey of definitions of life, right? This is one of those fun intellectual exercises. Can you define life? And I read lots of different papers. The one that really stuck with me was from Walker and Davies 2013, and we heard from Paul Davies earlier this morning, and Sara Walker is our next speaker. And so what they wrote in their 2013 paper is in the top blue box there. They say the transition from non-life to life is a fundamental shift in the causal structure of the system. Specifically, a transition to a state in which algorithmic information gains direct, context-dependent causal efficacy over matter. And so more simply, I think you can say life is when information takes control of matter, and I should probably add when it also energy. When information takes control of matter and energy, directs the system, determines the entropy, right, the probability of the future state of the system to very high likelihood. So the origin of life requires a molecular assembly which encodes information and uses that information to determine the future state of the system. By far, the most common and accepted idea for the origin of life is called the RNA world hypothesis. So RNA is ribonucleic acid. It's functioning in cells. It's best known for its role in transferring information, right? The central dogma of molecular biology is you have your genes in the DNA. There is a set of proteins that come in and they read off that information in the DNA and they transcribe it into a strip of RNA, which then takes it out to a ribosome, which reads it and builds the protein. So it's important because it is an information-bearing molecule. It is capable of storing and processing information. It also, the reason that it plays such a prominent role in theories for the origin of life is that it also can serve as a catalyst, very much like protein enzymes do. So a catalyst is something that enhances the reaction rate of a chemical reaction. RNA does okay at this. It can speed up reactions by a factor of a few compared to protein enzymes, which are highly specialized and can speed up reactions by a factor of 10,000 or so. And then there's been lots of really interesting experiments in the laboratory using RNA with help, and I've been reading some of these papers as well, and help usually means bringing in some of the machinery from E. coli, our favorite prokaryotic laboratory organism. But once you get RNA into a system, it's been shown to replicate, it evolves, it bifurcates, you get predator RNA, you get prey RNA, it interacts with its environment. So there's a strong case that RNA was involved very early on in the origin of life. Clearly you needed it to show up eventually because it's central to life today. You may be familiar with it from recent news where the Pfizer and Moderna vaccines for COVID are RNA. Basically, they came up with the RNA sequence that codes the protein, the spike protein on the virus, and then they found a way to get it through your cell membrane into your cell. Your ribosomes don't care if you made the RNA or somebody else made the RNA. It just grabs the RNA, reads it, makes the protein, and then since it's a spike protein for the virus, your immune system recognizes it. But I think that also shows some of the challenges with what RNA can do. So we are definitely going to go after the RNA world with our program. You know, we're going to look at the components of RNA. You can see the four bases there, purines and pyrimidines, adenine, guanine, cytosine, and uracil. They're relatively simple molecules. You can see they're ring structure molecules, or they have the 6-sided rings and the 5-sided rings. And then they have the backbone, so we can take a closer look at that on the next chart here. And the backbone is phosphate, ribose, which is a sugar, repeated. So you see the phosphate in red there, that's phosphorus, surrounded by 4 oxygen. Ribose is that sugar molecule there in blue, and those link up to each other over and over again. So they don't contain any information, they simply provide the structural support, and all of the information is encoded in those purine and pyrimidine bases. The main diagram there is for DNA, which is very similar to RNA. It has a different sugar. It has deoxyribose as opposed to ribose, and it has uracil instead of thymine. So there's some interesting changes in chemistry there. RNA is slightly different. So we're going to look for all of those. I actually did work on the origin of life early on in my career here at the University of Arizona back in 2004 with my graduate student Matt Pasek. Just to give you a sense of where origin of life research is, we didn't know where phosphorus came from. How do you get phosphorus out of a rock and into a molecule like this? Phosphate minerals like apatites, things that kind of make up our bones and our teeth, but are also very common in igneous rocks. They're totally inert, and they're insoluble in water. They're very common in magmatic systems, but there's no way for them to be reactive. So we actually looked at phosphorus minerals in meteorites because there's a wide range of oxidation states there. And we found that there's phosphorus in metal, and so a very low oxidation state, 0 oxidation state. And we did a simple experiment where we took that metallic phosphorus mineral, bonded with iron, and we dropped it in deionized water. And it reacted like crazy, and we got all kinds of a range of oxidation states of phosphorus, and it was lots of radicals, lots of interesting chemistry, long chains, pyruvates, and things like that. And that's still today considered the most likely source for getting phosphorus into these molecules. So that's the state of origin of life research. We don't know how you produce these purines. We don't know how you produce the pyrimidines abiotically, right? There's all kinds of metabolic pathways for producing these. We don't know how you produce the sugar. And, you know, a lot of origin of life research is just focused on making one of those phosphorylated nucleic acids, the phosphorus sugar base pair. That's where we are. We're trying to figure out that basic level of chemistry. We published that paper in 2004, and the community's still trying to take that forward to produce a molecule like this. There's been some success in that, but with very, very low yield rates. So there's a lot of critics of the RNA world. In fact, in the Shapiro paper that I cite here, they call it the prebiotic chemist's nightmare to describe, you know, how could you get something like self-replicating RNA? Going back to the vaccine, you know, I had to ship at very cold temperatures. It's a very unstable molecule. It's not meant in your body to last very long because it needs to get those proteins synthesized, and then it kind of needs to fall apart so you can rebuild different RNA strands to make the next protein that the cell needs. They're fairly complicated. They contain 9 or 10 carbon atoms, lots of nitrogen and oxygen atoms. You got to get that phosphate in there, and they got to be connected in a very precise 3-dimensional pattern. And there's lots of different ways to make those connections using thousands of possible nucleotides. And then that's dwarfed by the hundreds of thousands of stable organic molecules of similar size and composition. So somehow life had to take all of that complex chemistry, or you had to have an inorganic system that isolated and then polymerized this set of molecules. And we still can't demonstrate self-replication without that E. coli system coming in. So even in the lab with all of our amazing biochemistry, we still need life to come into the experimental system. We need that ATP, which is the energy molecule for all cells, or most cells. And even in the labs, when we know what we want the product to be with all of our chemical synthesis capabilities, we can't make this right now in the lab. So it's not clear how that happens. It's also not clear how the information can originate, because this would be a random process, but in order to undergo evolution and optimization through natural selection, there's got to be information encoded in the molecule, and it's not obvious at all how that would occur. So this is where astro-consciousness and the orchestrated objective reduction ideas really got my attention, because I just feel like we're missing something. I call it the spark of life. It's been used throughout literature since Charles Darwin era, trying to understand this problem. And so I started surveying all these different papers with different ideas about how the origin of life might have occurred. And then I read the Hameroff 2017 paper, Quantum Origin of Life. And I loved it. I just thought it was brilliant. I thought there was lots of great ideas. But what I liked most about it was it got down to the molecular level with very testable hypotheses, which is exactly what I was looking for. There's lots of ideas about the origin of life, but I can't test them with my asteroid sample, so they're really no use to me right now. So, and as a lot of this is Stuart's own words here, the RNA world hypothesis is driven by the principle that life is defined by a complex set of physicochemical processes, and when we're thinking about consciousness, that evolved well after life's origins, right? I think in the extreme case, there's people who say consciousness didn't arise until human brain and language evolved, which means that for most of the 4.5 billion year history of the solar system, there was no consciousness, and then it showed up with us. I think that kind of plays to the theme that Avi was talking about. That's very arrogant, right? To think that we're the only thing that's conscious and everything that came before us is not, and that makes us super special, so watch out, right? And so in the ORC-OR hypothesis, it proposes that consciousness preceded life, because we're looking for a motivation or a driving force, right? And so here they have that motivation. They're talking about the OR, right, the orchestrated reduction, which is self-collapse of the quantum wave function, which I understand is still undergoing investigation and needs to be demonstrated. It's theory, looking for experimental support right now. But this collapse of the quantum wave function, objective collapse of the quantum wave function, produces protoconscious moments that could have driven organic molecules to self-organize. And this is a figure right out of the paper where you've got these 6 carbon rings. They show how they could set up dipole oscillations in the pi electron clouds, and therefore they could enter the quantum superposition, and then if that undergoes OR collapse, that would be a protoconscious moment, and talks about feelings or pleasure and qualia arising right there in the universe, so you could start to see how that might be the motivation to say, hey, let's do that again, right? And let's get together and do it more often. So based on that paper, the first molecules to enter the quantum coherent state may have been what we call polyaromatic hydrocarbons, aromatic referring specifically to that 6-carbon ring. And these occur at the core of proteins, nucleic acids. You saw the ring molecules there, and of course the psychoactive compounds. That was the subject of many of the presentations yesterday. So when we take a look at the history of organic material, we can go all the way back to the interstellar medium, right? You've got a generation of stars that existed before the sun, and they created all kinds of elements through nucleosynthesis, and then they died either as a pulsing giant star or a supernova explosion, and they throw that material, the atoms, back out into space. And then you get into the interstellar medium, and they form giant molecular clouds, mostly hydrogen. This is stellar nursery. These are where stars are born. And the temperatures really drop in these. We get these cold, dense cloud cores down to 10 Kelvin or even lower. And you get ices forming, and the ices get irradiated. And ice isn't just water, but ice is methane, which has the carbon, or carbon monoxide, or carbon dioxide, or ammonia, which brings in the nitrogen. And through that UV, high-energy photon irradiation, you can start to make these complex polycyclic aromatic hydrocarbons. And then those molecular clouds collapse, and you get the proto-solar nebula, or what we call a protoplanetary disk. That image there, the orange kind of bullseye-looking target, is an actual protoplanetary disk imaged with the ALMA microwave array in South America. And so you can see there's gaps there, that the star at the center and those rings are, the dark rings are high-density regions where planets are forming. So we can look out into the galaxy and we can see planet-forming disks all over the place. And you start to get a lot more photochemistry because you've got higher temperatures and you've got that protostar forming at the center. And eventually those dust grains condense, collapse, and you start to get Fischer-Tropsch type reactions. Those are the FTT, or other catalysis types of reactions, and you can build up more complex PAHs, polyaromatic hydrocarbons. And then the dust grains coalesce and you start to form asteroids, and asteroid meaning mostly rocky metallic material, and comets, which also add some ice into that. And those have stayed intact since this time period. That's why they are ancient geologic samples, so we can go back and we can understand that entire history as recorded in the mineralogy and chemistry of the comets or the asteroids. Some of them got really big, big being 100 kilometers or so. They had radioactive isotopes, our favorites like potassium and uranium, which are still decaying in the Earth today, but also some things that are long decayed away, like aluminum-26 and iron-60. If you had ices during that exposure to the radiation, they would melt, they would start to aqueously alter the system, so you start to get other kinds of organic synthesis reactions like Strecker synthesis that can start to create different kinds of molecules. And nowadays, and actually since the formation of the planet Earth, those objects continue to tumble in from the main asteroid belt or from the Oort cloud where the comets are, and they land on Earth as meteorites. And that's what I did a lot in my career, early career, was studying those meteorites. So talking with Stuart, we started a collaboration. I was like, what should we look for? And we keyed in on his the transient absorption spectroscopy experiments he's doing with the group at Princeton and Greg Scholl's lab. And I said, well, we have lots of these really interesting molecules. Here's an example of the molecular structure of a complex macromolecular material that we find in meteorites. And you can see you've got all kinds of the aromatic rings, but you've also got some of the indoles and other nitrogen-bearing compounds in there. We have a synthetic version of this that we use for technique development because it's actually fairly intensive to isolate this from a meteorite. We're going to be testing that. We've already delivered the synthetic material to the Princeton lab, and so we're anxiously looking forward to that result soon. And then the other area that we're interested in is the amino acids. Now we're thinking about the building blocks of proteins. There you can see a picture of Stanley Miller, the famous Miller-Urey experiment back in the 1960s, where they were able to create what they thought was the composition of the primitive Earth atmosphere, do spark discharge to simulate lightning, and they produced a lot of amino acids. And the basic form of amino acid is shown there in the upper right. In the yellow, the amino group, which like ammonia is nitrogen and hydrogen, the carboxylic acid group in purple, which is the COOH. You've got the alpha carbon at the center, and then you've got the variable side chain R there. So these are way simpler than the components of RNA or DNA. Some of them contain as few as 6 or less atoms. What's interesting, when we look at the meteorites, we find 90 plus amino acids in meteorites, even though only 20 or so are used in biology. So when we look at these amino acids, you can see there's actually 21 here. There's an interesting one that uses selenium instead of sulfur there. So we're looking for these amino acids in our meteorite samples, and then we really want to understand how these could get together. So in order to make a protein, you need to start to polymerize the amino acids. You need to form the peptide bond, which you can see the arrow there pointing to the kind of the green highlighted area. So we got to start stacking these amino acids up, and that's what a protein is. It's a chain of these very simple molecules, and then they fold up into different ways to do catalysis and provide structural support, etc. One of the interesting things is that they're chiral. They have a left-handed and right-handed version. Life only uses the left-handed version. This is one of the biggest mysteries in the origin of life. If you produce amino acids through an equilibrium process, you're going to get equal amounts of left and right-handed molecules. But life only uses the left-handed version. I had some conversations just this morning on some ideas on how we might be able to do this using polarized starlight. That's a really interesting idea, and I'm anxious to learn more about it. But what I really got excited about continuing to read the Hameroff and Penrose literature was the way the information is in our brains. In their theory, there's a specific protein that plays a central role here called tubulin. You've probably heard a lot about it here. There's 1 billion tubulins per neuron or per cell here. And in the Hameroff and Penrose ORC-OR hypothesis, these molecules are the center of consciousness, and they may be central to information storage. This may be where our memories are stored, and so they have the ability to maybe do information storage and information processing. And so if we look at which amino acids are central to microtubules, we're looking particularly at tryptophan. I thought that was fascinating. There was a lot of discussions on the Monday workshop about the importance of tryptophan in establishing quantum superposition in these molecules. So that is what we're kind of keying off of. You see next to tryptophan, I've got it in the green box there, there's 2 other aromatic amino acids, phenylalanine and tyrosine. And I had a very embarrassing moment. So I have a bunch of team members. I don't do the chemistry myself for the organics on the sample. That's based mostly at NASA, Goddard Space Flight Center, and Johnson Space Center. And I said, hey, has anybody looked for these aromatic amino acids in meteorites? And they wrote me back and they say, yeah, you did 12 years ago in your PNAS paper with Sandra Pizzarello. So sure enough, it wasn't the focus of the paper, and I didn't even understand the significance of these amino acids, but we detected proline, which is not an aromatic, but you can see its form there. It does have a ring molecule. It's actually a little different than the other ones. But we detected phenylalanine and tyrosine, and you can see they're very similar chemistry. There's only that side chain, that OH, that makes the difference there. So these two can come in on meteorites. We've already shown that. We don't usually look for amino acids that are used biologically in meteorites, which is why I didn't remember we had done it, because they're often contaminated with biology. The microbes love these things. But I started to poke at this a little bit and try to understand the origin of tubulin. I started looking, do prokaryotes, those basic cells, have tubulin? It turns out that they don't, but there's a good evolutionary sequence for them. And I keyed in on this Ludenia et al. Paper here. You can see the B tub A and the B tub B. Those are the ones that are used in the microtubules. And then at the bottom of this evolutionary diagram, you've got the latest common FTSZ tubulin ancestor. So there's this FTSZ protein that is what is in the prokaryotic cells. It's the filamenting, temperature-sensitive mutant Z protein. And it has a fascinating form. It's involved in cellular division. You can see it's fluorescing here in this image on the left. So when a prokaryotic cell gets ready to divide, this FTSZ protein forms the filament, forms a ring on the center of the cell wall, and then it squeezes in and invaginates and ultimately separates the cell into 2 daughter cells. So it's got to be very primitive protein because cell division is central to evolution. You need a cell to make a cell, right? So I took a look real quick. I'm not an expert on this, but I said, okay, what in terms of these aromatic amino acids, I looked at the FTSZ. I went to the protein data bank. I downloaded the amino acid composition. Sure enough, FTSZ has the phenylalanine, but it's missing tyrosine and it's missing tryptophan, whereas the tubulin A and B both have tyrosine and tryptophan in them. So I'm like, okay, that's interesting. Maybe there's something in the evolutionary history. And of course, the microtubules do a lot more than just give rise to consciousness in our neurons. They're present in all eukaryotic cells, more advanced cellular life forms. They form the cytoskeleton. The cytoskeleton does all kinds of things like participate in mitosis, move nutrients and proteins around, anchor those organelles in the cell interior. So then I went to the literature to see where is tryptophan showing up. This is a bit of a complicated diagram, but we've got a range of amino acids. All the black dots are the ones in meteorites. All the green ones are the ones in biology. And then this is the highest occupied molecular orbital and the lowest unoccupied molecular orbital energy gap. And the ones on the left side in the green, those are thought to be the primitive amino acids that were used in the first proteins. And then through evolutionary time, additional amino acids were incorporated. The second to last one to get incorporated into proteins is tryptophan. And they think it's correlated with the rise of molecular oxygen. The very last one is that selenium-bearing one. That's an interesting story I haven't really poked into yet. But it looks like molecular oxygen in the atmosphere triggered the fixation of tyrosine and therefore got incorporated into the genetic code. And so if we compare the history of the atmosphere of the Earth, we see the first eukaryotic cells showing up right around the time molecular oxygen starts to rise in our atmosphere. Tryptophan became stable. If we want to speculate a little bit, that because they became conscious, that's what led to the complex cellular structure, and then, of course, to the first multicellular organisms about 1 billion years after that, as oxygen overtook carbon dioxide as the main oxygen-bearing molecule. So that's a speculation right now, but I find it interesting that tryptophan, which is the central amino acid in the ORC-OR area of investigation right now, is not present in the prokaryotic filamentary protein, but is in the tubulin. And so we're going to be going after that in more detail as we dive into the theories for the origin of life. And so get ready. Earth return of those samples is September of 2023. And thank you for your attention. We do questions at the end? We're going to do questions at the end of the whole session?
Can I ask if the next speaker's available? Yes? Okay, we'll hold questions at the end. And if we can, I'd like to introduce our next speaker. I don't see her yet, but
Hi.
We hear you, Sara. Oh, there you are.
Excellent. Okay, great.
Pardon me? She's ready. Okay. I don't have the title of your talk, maybe you'll, in front of me. The origin of life with information, something to do with that. And Sara Walker is at Arizona State University. She's a colleague of Paul Davies, whom we heard this morning. And so without further ado, Sara Walker.
Thank you. Can everyone hear me all right? Okay. I'm going to take that as a yes. I haven't heard no. So great. Thank you so much for having me. I'm so sorry I can't be there in person. I was really looking forward to meeting with old friends and new friends, but I unfortunately wasn't able to come down today. So this talk is actually kind of working maybe from the opposite direction of a lot of the content of this meeting, which is to start from thinking about the problem of life itself as fundamental and what kind of physics can we actually derive to explain the phenomena of life, and then how does that relate to other open problems, which I'll speculate about a little bit in the end. So I think we're all really interested in two fundamental problems that are at the frontier of science. One of them is the origin of life, which is the topic of this session, and obviously the theme of the whole conference is the nature of consciousness. And so I've been interested for a long time in the relationship between these two problems. And we're all pretty familiar with the idea that there's a hard problem associated with consciousness. And I was very happy that Dante introduced some of the ideas that Paul and I have developed over the years in the last talk, because one of the proposals that we had is really when we're approaching the problem of life, we should be thinking about an analogously hard problem. It's not hard in exactly the same ways as the nature of our experience is hard, which poses some challenges directly to even what it is to ask a scientific question. But I think life actually does challenge the way we do science in slightly different ways, and ways that are equally deep and fundamental. And the hard problem of life is actually exactly what we articulated as being the defining feature of life, which is that somehow in living systems, information, which seems to be an abstract property and kind of decoupled from physical stuff, actually seems to play a causal role in physical systems. And so if we talk about abstractions like mathematics or information, and they seem to actually be playing a role in the dynamics of physical systems, that seems like something outside of current physics. And so this is what we proposed was as the hard problem of life. And it's interesting to draw parallels between sort of current approaches to consciousness based on the idea of looking at neural correlates of consciousness. So we can't actually measure consciousness maybe directly with current theories and frameworks. So let's go after correlates of consciousness like patterns of brain states and things that we might associate with sleep or wake states. When we're defining life, we also do something where we try to define it in terms of the substrate level properties. And so I often think of these as the chemical correlates of life. So a good example is maybe for life on Earth, RNA. RNA is a specific molecule that evolved on our planet. We don't know if it's a general substrate for living systems. And what we need to do is figure out what are the more abstract properties associated with the kinds of structures that have evolved on our planet that we might be able to generalize to life anywhere in the universe. So we want to move away from focusing on chemical correlates of life and understanding more sort of the software or informational layer. So I think this quote from Roger Penrose is brilliant. I actually heard it on Lex's podcast, but I don't like the word materials because it suggests what we know what the material is. So one of the sort of issues with thinking about the problems of consciousness and life is that we have some intrinsic understanding that we understand matter very well, and therefore we should take our theories of matter and try to pigeonhole life and consciousness into current understanding of material reality. And perhaps the real problem is that we don't really understand a lot of the fundamental nature of, like what even the substrates are. And so I have spent most of my career really thinking on this idea that life might be explained by new laws of physics. And so Erwin Schrödinger obviously wrote this very famous book, What is Life? And so this turn of phrase, life is what, is actually kind of building on a quote that he had at the end of his book where he suggested that life might be governed by other laws of physics that we don't know yet. And I'm always kind of perplexed by the idea that people want to take physical theories that have been developed to study very different phenomena from life and mind and somehow expect them to apply to life and mind. Whereas if you look at the history, the progress of theoretical physics, almost all of the revolutionary understanding that we've ever got was actually looking at a phenomena for that phenomena and building theories from that phenomena. And then we worry about unifications later, which is one of the reasons that we're having a problem trying to unify gravity and quantum mechanics right now, because they were developed to describe completely different sectors of physical reality. And so part of the reason that I think there is a strong argument that there might be new laws of physics that we haven't uncovered that apply specifically to life and potentially also to mind is that we haven't actually really done the hard work yet of starting from first principles of thinking about those phenomena as fundamental and building theories based on the properties of those phenomena. And I think there is a lot of that momentum being built across different communities right now, but I think that's where the problem stands, is that there are no theories that were specifically developed for those phenomena that are supposed to be at the level of description of our current most fundamental understanding of nature. So where I think we sort of sit being trained as a theoretical physicist and looking at the history of physics so far is that we've had all of these unifications of seemingly disparate phenomena in the history of our understanding of reality that have given us a much deeper picture of what's actually going on. So some very prominent ones are obviously the unification of space and time by Einstein and Minkowski. One of my favorites is the unification of terrestrial and celestial physics. So, before Galileo and Newton, no one had any idea that we could explain motions in the heavens by the same principles that we could explain on Earth. And that was actually, it seems obvious to us now because we've been taught that for generations, but that's a huge conceptual leap to assume that all motion can be described by a few unified equations. I mean, it took a lot of progress of human thought to get there. We had to invent geometry, we had to invent measuring devices for time, we had to get more accurate and be able to measure seconds, which we hadn't been able to do for most of human history, to even get to the point that we could actually propose and test those kind of theories. And in the last century, we did something very interesting, which is we tried to build theories of computation based on the abstract process of the way we think our own minds work. And that was obviously started most prominently by Alan Turing, trying to think about formalizing the function of what it is to think. But those kinds of theories of computation that we have now have not actually been unified in a really rigorous way with the theories that we have of matter. And so part of the conjecture I have about the physics that will solve the origin of life is actually to think about what is the deeper theory of physics that explains both the concepts that we talk about as information and computation, and also the concept of what it is to be a material object, what it is to be a thing that physically exists in our universe. And unifying those will probably not look at all like either of those things look now, in the same sense that almost all of these unifications, if you think about those concepts before and after we made the unification, the concepts looked entirely different on either side of that intellectual transition. And so really the sort of thought exercise we need to go through with understanding the transition to the origin of life and what physics might govern that is to imagine what's possible to explain the phenomena we call life with physical principles that might be beyond sort of standard physics and what we know right now. And so when I think about this problem and thinking about the nature of information, I think it's very clear in some examples that some kind of concept of the word that we use, information, is going to be absolutely critical to explaining some phenomena in our universe and why they exist out of this seemingly infinite space of possibilities of things that could exist. And so these are just two of my favorite examples. They're very anthropocentric, but I think it's because minds are obviously the most prominent examples of the physics of life that we have. And they're also the ones that we're most intimately familiar with because we are minds that exist in the physical world. So these two examples are really meant to demonstrate the fact that when you have physical systems that evolve certain kinds of knowledge, gravity in the left-hand case, or knowledge of nuclear physics in the right-hand case, that knowledge actually becomes important to explaining certain features. So for example, we would not have thousands of satellites orbiting our planet right now unless we had a biosphere emerge early in the evolution of our planet, and that biosphere evolved intelligent agents like us that eventually came to understand regularities in the physical world that we call the laws of gravitation that allowed us to build things like satellites and launch them into space. So the existence of satellites orbiting a planet in the way that we have it with modern technology, the causal chain of events requires some level of abstraction in order for those things even to exist. And the same thing with certain elements in the periodic table. If you think about all the physical processes that generate the different elements, some of those are very obvious physical processes like Big Bang fusion or the death of stars. But technology, if you look at the highest atomic number elements, is the only process in the universe that can reliably produce high atomic number elements in high abundance. So for example, element 118, magnesium, which is the highest one that we synthesized in the lab, can be produced under tightly regulated conditions. So if you have an intelligent agent that has knowledge of nuclear physics and can build boundary conditions to control the setup and build all the constraints into the universe to produce magnesium, can do it repeatedly and can do it as much as resources and energetic constraints would allow. So it's a process that's mediated by the evolution of technology in our universe. So explaining those specific features of what we observe on our planet today require information to be a part of the story. And so this gets into something very interesting for me, being a theoretical physicist, thinking about what mathematics is as fundamental to the nature of reality, because it seems the fact that our minds can abstract things then becomes causal to things that actually are produced in our universe. And that causal chain of events, that this thing happened and then this thing happened, is actually really critical to that structure. But most of the time when we think about the nature of existence, we have a very hard time with dealing with why some things exist and why other things don't exist. And this is most prominent when we're thinking about, for example, why the laws of physics in our universe look the way they do. And so some people, probably most prominently Max Tegmark, which is why I'm citing his mathematical universe hypothesis here, have proposed that perhaps all of physical reality is just mathematical structures. Well, not just, I mean, that's a very elegant and deep idea, but that all possible mathematical structures exist somewhere. And these kind of proposals are very elegant because they basically give everything existence, but then you have a hard time explaining selection and the existence of things in our particular universe. And so one of the reasons I'm bringing this up is because this becomes very deeply related to the nature of life and what information is doing in our universe, because the process of acquiring information is basically a process of selecting what gets to exist and what doesn't. That's what natural selection is. It's a filter for what gets to exist. But Darwin's theory is not sufficiently deep to actually embed that in the fundamental structure of physics. So if you take that intuition and then you ask questions about what is the nature of reality when we think that not all things that can exist do exist and some things get to exist and the things that exist actually cause what gets to exist next, that's really the major question about understanding information as a physical thing, because information seems to play the most critical role in who or what decides what exists. And so so I've already connected this a little bit to Darwin's theory, but I think this is really interesting about where the dichotomy is between how we need to explain life and what we have so far in the past 300 years of theoretical physics since it was invented by Isaac Newton. So Isaac Newton had this conception of laws of physics that they existed autonomous to the universe and they exist in some sense outside the universe. And that introduces a whole host of problems because you have to ask where the laws of physics come from, and you also have to specify an initial condition. Darwin had this very different conception of the way the rules of the universe behave, in the sense that he thought that there were endless forms most beautiful and wonderful being evolved as a contradistinction to the idea of fixed laws. So in some sense, he was implying that the laws themselves or the rules of what gets generated next should change in time. And a lot of people have actually talked about the idea that the laws of physics should change. Paul Davies, that has been mentioned a few times and spoke this morning, and is one of my mentors, and I look up to him quite a bit, has proposed that. And I've worked on that kind of idea with him. Lee Smolin has also proposed the idea that the laws of physics themselves might change in time. But this is really sort of one of the problems you're confronted with when you try to look at biology from the perspective of fundamental physics, is the idea of fixed laws don't seem to fit. And I want to relate this to a particular issue we have when we're trying to think about definitions of life. So there's the sort of dichotomy between how current physics works and how we might come to understand life. But there's also sort of these definitional approaches, going back to that idea of chemical correlates of life that I mentioned at the beginning of the talk, that we can just go in and kind of superficially define life based on the physical structures we observe. And I like to make the analogy, it's a little bit like defining gravity by studying the properties of rocks. Like the level of abstraction to understand that gravity is actually what's binding, rocks together when planetary formation is happening is an exceptional level of abstraction, and it required us hundreds of years to develop that kind of idea. Right now, we have this sort of conception that the fundamental unit of life is the cell, and we want to define life in terms of self-reproduction, because cells can copy themselves and they can evolve and they metabolize, and all of those kind of words become part of the standard canonical definition of life. But if you start to reframe the question of life from the perspective of thinking about these more fundamental abstract ideas, that information is really important. It's about how information propagates in matter, and that actually leads to the structure of selection and the structure of the possibility space that our planet happens to be exploring. The fundamental unit of life is not the cell, it's actually the propagating lineages of information. And in the theory I'm going to talk about in a few minutes, we call those causal chains. Basically, it's the idea that when you build a structure, that structure becomes a necessary precondition for another structure, and that actually propagates in time. So I myself, for example, am not I'm not my atoms, and I am actually an informational pattern that happens to be propagating in a particular set of atoms. So I'm not the same atoms I was, even this morning, because I had breakfast and I had lunch, but I am the same Sara in some sense, that I'm a coherent structure that maintains myself, and I am constantly imprinting myself into matter. I mean, if you think about that beyond an individual, of course, I inherited information from my mother. I inherited information culturally from the societies I live in. So I'm actually just a local aggregate of propagating bits of information. So when we're asking for this sort of idea of new laws of physics that might explain what life is, these have to try to explain why it is that these certain lineages of propagating information exist, and how they select out basically what exists from what doesn't exist. And I think the real challenge there is that we have to move into a domain of physics that we haven't entered previously. And so if you look at the history of physics, we have always started by trying to explain some feature over many centuries that we slowly began to resolve. And I think, thinking about space-time is a good example, because in ancient times, the reason that we invented geometry was because we were trying to map the physical landscape. Geometry literally means measuring Earth. And we were trying to, look and try to measure the height of mountains and things. And then we abstracted ideas like triangles and circles. And over the centuries, we could use that knowledge to actually be able to build theories of planetary motion, and then the concept of space-time itself being geometric. One of the things we haven't done yet, and if you think about it, Newton was an alchemist. Chemistry was magic for him. He didn't actually do physics of chemistry. His contemporary Leibniz tried to, with his monadology, to actually regularize biology into kind of an abstract set of principles, but it was too early in our history to be able to do that. We didn't have a deep enough knowledge of chemistry. So what my point is with this is you look at the history of where we've actually uncovered fundamental laws of physics. They're in primarily three sectors so far. We have laws that govern the properties of matter and light. So the most, fundamental structures of matter, the very simple bits, that the atoms and elementary particles. We have laws of motion and simultaneity that allow us to talk about space and coordinates in space and how space behaves. And we have laws of large numbers and uncertainty, which are the thermodynamics we developed in the last century to explain certain properties associated with the flows of energy and work. So one of the questions with this idea of thinking about the physics of life and the kind of unification I'm proposing about information and matter and all these ideas is obviously we don't know what we're talking about yet because we haven't invented that physics. But that physics has to somehow sit. It emerges in chemistry because life emerges in chemistry. And so the conjecture I have is that it's really about the physics of what's possible, because chemistry is the first scale of physical reality. If you're thinking about elementary particles, building atoms, building the periodic table, and then the combinatorial diversity that emerges in chemistry. Chemistry is the first scale of reality where everything that could exist doesn't get to exist. And so there have to be new principles that emerge there to tell you why certain things exist in our universe, and we just don't have an explosion in a Tegmarkian sense of all existence existing all at once. And so the theory I work on that's trying to address that is assembly theory, but I just want to give a little bit of a nod to other theories that I think are being developed right now that I think are in a similar space. One of them, I think, which is closest to assembly theory, are causal set theories of quantum gravity. There's also constructor theory, which has really inspired me a lot in my career. And I think the Wolfram Physics Project also has a lot of parallels in terms of trying to define reality in terms of hypergraphs, which have a very similar kind of structure. I think the difference between assembly theory and those other theories is what we're doing in assembly theory is explicitly trying to explain the phenomena of life and start from empirical data that we can get in the lab and constantly make contact between how we think about the space of what's possible, how to define it in terms of a fundamental physical structure, and iterate that between what we can measure in the lab. And use that to design original life experiments. Because basically what we're trying to look for is the transition of the original life being a transition to where information has to be a part of the narrative of your physics. And instead of talking about information, we talk about assembly, because assembly is actually a physical instantiation of what information is. So this is sort of the broad sweeps proposal, is that the original life is actually a problem of unification, matter, and information, and that's going to look quite different. And before I get to explaining a little bit about what we're doing with assembly theory, I just wanted to give one more little provocative thing, because I tweeted this morning, and it just happened to be on my mind, which is that matter and information equals time, because we had this session on time this morning. So whatever physics that you want to propose that might explain the phenomena of life, in some limit has to reduce to current physics. And I think when we talk about assembly, we have limits that reduce to entropy, and we have limits that reduce to sort of standard physics. But those are the no information limits in some sense. And they also maybe recover some of the features of current physics, where time is not really a fundamental feature of the objects that exist. And the proposal in assembly theory is that I am not actually an instantaneous object that exists in the universe. I know Paul mentioned, like Paul has a mutual information with his past self. I think that's the best way we can describe it in current physics. What we would say in assembly theory is that Paul or I is an object extended over time. I didn't just appear in the universe. I actually require an evolutionary lineage of events. And that lineage of events is actually part of my causal structure as I exist in the universe. And this is what we actually are trying to do in assembly theory. So I'm going to introduce sort of the basic idea. As imagine you're thinking about the space of all possible things. We can talk about that pretty easily in mathematics. In physical reality, it ends up being quite hard. So if you want to think, for example, of defining the space of all possible technologies or all possible minds, That seems like a very intractable mathematical task. But if you think about chemistry, molecules are combinatorial objects. Iterating over the space of all possible molecules is actually something that, at least in principle, you can think of algorithms that could do. And we do have this in cheminformatics. And so one of the things I mentioned already about why it might be possible that new physics actually emerges in chemistry is that chemical space is vast. So I made this point already, but I want to make it a little bit more concrete. When you get to the space of all possible molecules, the combinatorial diversity is so large that not all possible versions of a single molecule can exist. So the molecule I've shown here is Taxol. This is a slide that I borrowed from my collaborator on assembly theory, Lee Cronin, who's worked from his lab I'm going to show in a moment. But if you look at all possible combinations of the set of atoms, the volume of resources you would need to make every single iteration, every single stereoisomer, every single thing with a consistent molecular formula is huge. And Stuart Kaufman has always made these kind of arguments in terms of thinking about proteins. If you think about a protein of length 200, it has 20 amino acids. There are not enough resources in the universe to make every possible variation of that. And so there's some interesting features that emerge when you get into that space about trying to think about why these particular things exist, and also the adjacent possible space of things that could almost exist, and then the space of all possible things. And this is really what assembly theory is formalizing. So to get to the sort of core idea, the fundamental object that's easiest to think about with this proposal of assembly theory as an explanation for what life is a molecule. And the reason is, for all the reasons I've stated, that chemistry is a first scale reality. This physics becomes apparent. Molecules are easy to describe. They're also easy to empirically measure features of in the lab. And so assembly theory is based on the idea, if you imagine a molecule and you break it apart and you break it apart again, you can get fundamental building blocks for that if you break it all the way down to elementary building blocks. And in assembly theory, as it applies to molecules, those fundamental building blocks are bonds. So now you take those bonds that are present in a molecule like ATP, for example, and you think of all the ways that you can build that molecule back up. So those are what we call causal chains or assembly pathways. Those are directed paths where you recursively use pieces you've generated before to assemble the molecule. And that forms a causal graph of all the ways that you can assemble that molecule from elementary building blocks. And the larger the molecule, the more, the less symmetry it has, the larger that number of pathways to assemble it is, and the deeper those paths are, the longer they are. We quantify a feature of that assembly space as the shortest path in that space as a measure of how much time or how much information the universe needs to have in order to produce that molecule. It's sort of a minimal bound on how many steps you need to do to produce that particular molecule. And the key conjecture of assembly theory is that this minimal path is related to the physics of life, because if you're imagining the combinatorial explosion of all chemistry, the further you get into that space, the longer the assembly path, the larger and larger the combinatorial space of what's possible. So if you were doing a random walk, you would never hit that molecule. It would be such a low probability event, and you certainly would never hit that molecule twice. You would never hit it 3 times, and you would never ever hit it a mole times, or 10,000 times, enough to pick it up on an instrument to actually measure the existence of that molecule. So there's an argument here that if you observe molecules with a sufficiently long assembly index, it indicates that there had to be a machine or a information processing system or a system that's selected for the existence of that particular molecule. And so assembly index basically becomes a ranking, an ordering of all the possible molecules that can exist based on how much time or how much information is necessary to already exist in the universe to produce it. So the information is necessary to specify specific steps to produce that molecule. Now, an important feature of this is that it's actually measurable in the lab. So a lot of sort of conjectured theories that want to propose reality is fundamentally about causal graphs, and the causal relationship between events have yet to connect with observables. But what we're doing with assembly theory, because we're starting with the physical objects of molecules themselves and deconstructing them into causal graphs and talking about the properties of those graphs, it's in principle possible to measure features of that in the lab. And so this is really the genius of Lee Cronin, my collaborator who came up with the idea of molecular assembly and molecules in the first place, is that you can actually measure the molecular assembly index directly from mass spec. So if you take a mass spec, it fragments a molecule into pieces, and the fragmentation pattern in the mass spec is directly correlated with this number associated with how complex, quote unquote, the molecule is, or how assembled it is in the language we would say in assembly theory. And so this paper was published, I'm sorry I don't actually have the reference appearing on the slide here, but this paper was published in Nature Communications last year, and I'm happy to give anybody the reference if you can't find it, which was out of Lee Cronin's lab. And basically what we did was take a whole bunch of samples of non-living systems, living systems, recently deceased systems, blinded systems from NASA, and applied molecular assembly to those systems to try to determine if this conjecture that there should be some fundamental threshold in the universe where objects are so assembled you never observe them outside of living systems actually would stand up to empirical data. And so in fact, what we found is it does. And so if you look at a molecular assembly index of 15, so you take approximately 15 steps in this assembly space as the minimal number of steps to produce a molecule. Remember the ATP I showed actually had 21 as its minimal number of steps. Above 15, the only systems that we've observed so far that produce those molecules are biological systems or things derived from biological systems or technological systems. So obviously technology can produce high complexity molecules. And so 15 seems to be the threshold. And the way I think about this is relates back to what I was saying about sort of fundamental physics of life, that there's a scale of reality where this physics has to become apparent. Is if you think about the space of all possible things that could exist, what the assembly theory is saying, what this empirical data is suggesting, if you take 15 steps into that space or more than 15 steps, when you get to the level of chemistry, then you have to have selection and you have to have systems that have knowledge specific to producing those molecules. They have information in order for those molecules to exist in high abundance. Because without that, the statistical probability of ever observing them is so low, you should never observe them. Your instruments would never pick them up as individual objects. And so this is sort of the key empirical result so far. And what we're doing now, which is the part I'm much more excited about being a theorist, is actually building some of the ideas I introduced in the introduction into assembly theory. And we have a paper that will be hopefully pre-printed in the next couple of weeks. I was hoping it would be before today, actually, so I could point you guys to it. But I want to talk a little bit about why I'm excited about assembly theory as an explanatory framework for life and some of the deeper ideas connected to the physics I described at the beginning. Because in my field of astrobiology right now, there's a lot of misconceptions about what assembly actually is. So for example, people think it's just a complexity-based measure for molecules that tells you whether they might be alive or not. And there's some arguments that, for example, some geochemistry on some planet might be able to make a molecule that's higher assembly than 15. And the specific empirical value measured on Earth is not so much the critical feature. The fact that there is an empirical boundary is a critical feature, because basically what it says is if you're expecting the universe to spontaneously and randomly generate objects of very high complexity, that basically means that cooked into the laws of physics is design of those objects. So example is I would never expect my glass, I guess I always have a glass of water or something nearby, so I use these a lot as objects, but I would never expect this to spontaneously fluctuate into existence on the surface of Mars. It requires a long evolutionary process and also other objects to exist that are similar to it in the past in order for this to even be able to come into existence. That's sort of the key argument of assembly theory. But people still have this kind of idea in their mind that a complex molecule can appear for free or a Boltzmann brain can appear for free. Assembly theory says, no, those things can't appear for free. They require a causal chain, and the causal chain is actually a part of the structure of that object. And so what I'm showing on the right here is how assembly theory basically coarse grains physical reality by assembly index. What I mean by that is you start from fundamental building blocks. This assembly index basically tells you how far into the space of things that are possible you've gone to produce a particular object. And when we look at something like the evolution of biology on Earth, objects are ordered in this space by their assembly index, and that assembly space structure tells us the relationship to other objects, how much information they have in common, how much shared causal history. And then you can build up the entire sort of evolutionary pathway of a set of objects based on that common assembly space. So assembly has a really deep relationship with time because assembly objects that are more assembled require more time for their formation. But more fundamentally, the way I think about assembly spaces is an assembly space is basically saying ATP as a molecule is a structure that exists across time. And the easiest way for me to explain what I mean by that is to think of a stack of Legos, for example. Like you can imagine having a stack of blue and yellow Legos. Maybe you have 5 blue and 5 yellow, for example. Now take your stack apart to the 5 yellow and 5 blue blocks and think about all the ways of assembling it. In order to actually see the structure of the assembly space, you have to unfold that object across time. And so that's what assembly theory is saying, is that these things actually have structure that is temporal in extent. Elementary particles don't have this, molecules do. We certainly do because we're even more complex. And the biosphere's evolution is basically one of increasing assembliness, which means we're getting deeper into the space of what's possible, into that combinatorial space, which is being constrained by what's existed in the past. And so basically what we're doing right now is formalizing all of that into a theoretical structure. So this is just one image from the paper that I mentioned that we're working on, which basically says that most ways that we talk about things right now is just all things can exist. If you think about, say, for example, thermodynamics or structure forming systems, people put no causal constraints and no history into the formation of objects. They just assume that you can just randomly grab things and join them together and get some double exponentially growing space of possibilities. Assembly theory says you have to start from the objects you observe, and they co-constrain possible histories, and they also co-constrain possible futures. And this is sort of the causal cone of where we exist. I'm an assembly space, you're an assembly space, we have some overlap, so we share information and causal history, and the things that can be generated in the future are based on the structure of our past. And we can actually quantify those features. And so this is just to say like how this actually maps to thinking about biological evolution in terms of the structure of causal graphs and assembly spaces. We have basically seen, if we look at speciation over time, that we've had all of this novelty being generated out of this space, but in the past, there's, a common ancestry to that structure. And in this sort of abstract sort of causal diagram sense, we can look at all of these observed structures. When you actually look at the physical graphs for objects, for example, it doesn't have to be molecules. So I'm for example, applying, we're applying assembly to a lot of things like morphology and collective behavior and things between Lee's group and my group right now. But if you look at the graphs, you can actually talk about co-assembly spaces where, if I had two molecules and they share a common history and then it bifurcates off, that this structure actually is the key constraint for talking about why not all things exist. You exist locally in the space of what's possible, and there's causal constraints imposed by you as a causal structure that exists in that space, and your relationship to other things that you can be co-localized with. You can't exist, not all things can exist at the same time, and they certainly can't exist in a local space of all the things possible. So I actually don't know where I am on time. It's on Zoom, it's really hard to tell where you are. But I just wanted to go back to the first slide about talking about hard problems, because I know it's a conference on consciousness, and I've thought a lot about consciousness, not my main area of research. I'm really interested in solving the origin of life problem. And assembly is designed to do that, because basically what we're aiming to do is through this construction of this theory, be able to tell you the conditions the origin of life should emerge, and then go in the lab and test it by measuring the assembly.
Sarah, try to wrap up, please. We're running a little over.
I will.
Thank you.
So I just want to point out that I'm going to kind of skip through some of the consciousness stuff, actually. I want to just go to the end and say that there are a couple questions about the nature of consciousness that I'm particularly interested in, because people usually focus on the hard problem of experience. But I think that problem is hard for a lot of reasons that people here will be very familiar with. I think one of the things that illuminating thinking about the hard problem of life helps with is that maybe the focus should be on, if experience exists, does it have any causal consequences that if non-experiencing entities didn't exist, they wouldn't have? And so this for me is really interesting. It's also kind of the formulation that Dan Dennett had in terms of his hard question of consciousness. And then there's also this idea of whether there's future consciousness we can only measure across space and time, and whether they can only be measured at the level of collectives rather than individuals. So I also really like this idea of consciousness being contagious. It's not just a property of our individual minds, but there's something about the relationship between our minds. So these things are captured for me by thinking about this kind of thought experiment that we imagine rockets for centuries before we built them. So there is some kind of congruence between the things that we're imagining and we're thinking about and what actually happens in the physical world. And some people might deny that, but I think that's a real feature we have to explain. And in assembly, I think there are some connections that we could talk about, which have to do with the structure of the assembly space and how that relates to consciousness. But I think I'm going to stop there because it's been a lot of stuff introduced, and I just really want to have time to thank my group and my collaborators, and thank all of you for listening, and thank you.
Sara, thank you very much. You actually finished on time, and I appreciate that. We're going to hold questions as we did for Dante and for the three of you at the end, and so we'll thank you again, and it's my pleasure to introduce the third speaker, Steen Rasmussen, a professor of physics at the University of Southern Denmark, who's been working on life, artificial life, real life, and its origins for many, many years. And he'll be speaking on assembly and evolution of artificial living materials, the second origin of life. Please welcome Steen Rasmussen. Did anybody lose a cell phone over that part of the room? It's over here.
Yeah, thanks for having me here, Stuart. I want to sort of express my gratitude. This is not my area, consciousness. I'm still confused about what it is, but I've been lucky enough to participate in several of these meetings, and I have to say that these meetings have been some of the most inspiring periods of my scientific career. I've gotten lots of good ideas, and I've heard some of the most enlightening presentations I've ever heard. I mean, the last one we had here with Avid Loeb was really inspiring too. So Stuart is really doing, or has done over a million years, a really incredible work on keeping this thing going. So yeah, let's get going here. So I make sure that I introduce or acknowledge all these incredible people that has helped me. Without all this help of people that are much smarter than me, I would never have been able to present these things that I'm going to talk about now. The people in red are the ones that are sort of my most current collaborators, and I'll show some of the new results that hasn't been published yet. So I have a plan here. I'll talk about what life is. I will present to you my perspective on the possible origin of physical-chemical life, and then I'll focus on how we can create life in the laboratory. So this presentation is about how to make life from scratch by doing a demonstration, an example of a demonstration. So there are problems. It's not easy to create life, so I'll focus on what the remaining issues are. I'll say a few words about evolution, because I think there are some illusions out there about what evolution is. And then I will talk about the potential implications for once we've created artificial living materials, and of course how it relates to, in my opinion, cognition, sentience, and consciousness. So artificial life is man-made life. The difference is that we make it instead of it somehow emerged from without some intention. And my operational definition is that life is a physical process that is able to take in resources, turn the resources into building blocks, and then have these building blocks grow assemble and grow and divide. And then this growth and division has, at least in part, to be controlled by information that is inheritable. And if you allow this information, inheritable information, to change a little bit from one generation to the next, then you have selection and loss evolution, and then you are done. I believe it's a good way to think about these things is that there is a gray zone, that it makes sense to talk of life-likeness. I mean, that things can be more or less lifelike. And in artificial life, we study the limits on material systems that can support living processes, and a number of people, like myself, study wet carbon chemistry. Other people study robots, and also living processes in cyberspace, which I've done quite a few studies on too. And then also ecosystems that sort of mixes everything up. We're only going to talk about the first part. So the likely origin of life starts with a free energy gradient with some starting material, some reactants, and catalysis. And catalysis, just to refresh chemistry for some of you, that's probably a long time ago, a catalyst is something that can really speed up the process dramatically of certain processes while leaving others without any speed up. So at the core of a metabolism, any metabolism, you have catalysis, where you have resources that are turned into building blocks, and they can only, this catalysis also is very sensitive to what kind of environment you're in. So it can't occur in any environment. Now, one of the important things, I believe, is that this catalyst has to be able to catalyze itself. You call that autocatalysis. So that's a very important step, and it's not so difficult to make autocatalysis. Many systems, they have that. But the next step is a sort of a little bit of a mystery, because we have to require that this catalyst can be in different states, and that there should be a combinatorial variety of catalysts, and that there therefore should be the possibility for actually a selection of catalysts, so that you have a catalyst that produces catalysts that with a little bit different properties, and then you have, so to say, a catalyst that can hijack this transformation of resources, of the transformation of resources into building blocks. And I just want to emphasize one thing, that just to formalize a little bit, that you can actually quantify. Let's assume you have a catalyst with W different states, and this can actually encode information, and this is Boltzmann's equation. And you can define information in many different ways. The traditional one, when you think about biology, is information on a DNA strand or RNA strand. But that's not so important, I think, here. Information, there's also another notion of information that is compositional information, where it can be a composition of an aggregate where the sequence doesn't make sense. So a sequence of information is a particular kind of information. But compositional information is really the most important one. So Landau's principle, you can get that by applying fundamental thermodynamics to Boltzmann equation, and then you get that the most fundamental process of creating or changing or deleting one bit of information requires energy. So that means that you need to have free energy available. So that means that you need to have some kind of metabolic process running before you can actually generate information, before you can contain or save and store information. I think that's really important, and a lot of the discussion in the origins of life community focuses only on information, but you need to have this engine, and nothing moves without the available free energy. So now we are ready to talk about how to make such a machine in the lab here. We just need to make sure that these resources, they also make building blocks to a container that can keep together, that's able to keep together what's it called, this metabolic machinery with the catalyst. And if you have that, then, I mean, one way of looking at it is that you need to have a metabolism and this informational catalyst in this container or on this container. It has to be kept together in a particular environment, because obviously you can't use any kind of environment. So it should be noted that these component interactions generates the property of life. It's not that one molecule is alive, or it is simply, life is a process that has been generated out of or emerges out of these interactions. So this is the design of our protocell. Again, my goal for many years, since I was a young man and I met Stuart, where he was also a young man, it was simply to try to make an example of that. So we have a situation where we, on the right-hand side, we have resources and free energy that we feed to the metabolism. The metabolism then takes these resources in and create building blocks both for the informational system and for the container system. The container system then makes sure that the metabolic compound and the informational compounds, they can sit on the container, so they have been brought together. And then the informational system, remember that's the one that, it's this combinatorial system, it can catalyze the formation of these building blocks. And I want to say that the most important inspiration for me has been Manfred Eichen's idea of these coupled autocatalytic compounds, and Prigoghine's idea of free energy as being the driver for self-organization, and for Neumann's idea of actually, he believed back then that both living and intelligent processes could be carried by many different kind of substrates. So as we'll see here, we are also trying to use different substrates than modern life. Many people have come up with ideas for how to create sort of this first instantiation of life. As you've heard earlier, the most famous one is the RNA world, which is down in the left-hand corner. But we can also, we made some diagrams, a functional diagram language, because it's very difficult when you're working on very different types of chemistries and you have different designs, it's difficult to compare. I mean, who has actually solved which problem? So this is a platform we can use for that, and we've also used it for robotics, and you can also use it for processes in cyberspace. Just to emphasize that there are many groups that are working on different aspects of this. So early on, I had, or our teams have had sort of nightmares or dreams about what kind of material, what should the starting materials be? And there are many choices. And at the end of the day, it was clear that it's really hard, since it's really hard to get money to do this kind of work, to sponsor the chemists to do this and pay my own salary and so forth. We had to use robust, very simple, and very inexpensive chemicals that chemists were familiar with. So at the end of the day, as you'll see in a little while, we used, so to say, the ones you can go to the grocery store and buy. Or maybe not the grocery store, but the simple ones. I don't think these are the ones that were used in the origin of life, but I think it's still valuable to say, take simple building materials and see whether you can actually get these things to create a living process. Besides starting materials, we had to figure out how we should organize these things. And we came up with a simple design where the container is, you can think about that as a used piece of chewing gum, so it's kind of sticky. And you can then put the informational molecules and the metabolic molecules on top into them. So it's a sticky thing where these things, they are on the surface. So the container is exterior of actually, it's a vesicle, and these two types of aggregates, they are attached on the outside. And this is nice because then you can take up resources from the outside and you get rid of waste without any problems. You don't have all this monkey business with having a cell membrane and you have to transport things inside and get the waste out, and it just makes things much more complicated. And I think that there are nice pathways to actually go from this more primitive and simpler design to a modern cell later on. Okay, I know that I will not go through all the details here, but I want to show for those of you who are interested in chemistry that at the core of this design we have a metabolism, and it's a ruthenium tris by pyridine design. So we have a molecule that can capture light, and as it captures this photon, the ruthenium molecule kicks an the photon kicks an electron out to one of the antennas, and it sits out there for a while until it jumps back if we don't do anything with it. But this is an energy-rich electron that we can use to do chemical work. So what we do then is we make sure that we have an electron donor that can make the ruthenium atom happy, so then we can use this high energy electron for do chemical work. So that's actually the engine. So it's a very, very primitive kind of photosynthesis you can think of. And what we've demonstrated early on was that we can use this metabolism to generate building blocks both for the container, fatty acids, the canoic acid, as well as we can convert protected oligomers into functional oligomers, so you can ligate them and turn them into DNA plus and minus strands. And these are these were the first experimental evidence that you see here. So there's a little more details. These are also from some of our first experiments, where we start out with oil, oil-like substance, picolinate ester, and this oil-like substance this is just one of the metabolic processes. You can then turn this oil-like substance into fatty acid, and this is just a demonstration. We have sonicated oil droplets, and as you decorate these guys with ruthenium, you can then over time see the emergence of these vesicles. And if you don't have any metabolic process running, then you just end up by having all this oily substance, this picolinate ester, to sort of precipitate onto the glass. We've gone back to study these metabolic processes recently, because now we are ready to launch a new experimental adventure where we actually incorporate the electron donor into the DNA molecule. So these are just details, but about the reaction. This is a metabolic reaction network, and there are three interesting reaction constants here, and one of them is the what's it called, photoexcitation rate. So actually, this whole metabolic system is rate limited by how much free energy from light it can take in. So that's actually the bottleneck. Then there is some molecules that indicate the entropy death. I mean, when does things fall apart? And then there is also another, the green one, that is indicating how this quantum, charge transfer is happening. There is because we can modulate that by changing the catalyst, namely the compositional information of the DNA. So our first protocell life cycle without information replication was back in 2011, so it's more than 10 years ago. It was a PhD job by Sara, and what we did, we took fatty acid and we sonicated it and we put it through a filter, so we got small vesicles of more or less the same size, and then we were feeding the system with this oil, and the oil doesn't like water, so it goes into the membrane, and you got these big clumps, and then you apply light, and then you get division of these things, and you can then run this system until you get tired of it. People were criticizing us and saying, well, maybe it's not, you don't have inheritance of individuality, so we had to demonstrate that we indeed do have budding, and you'll have sort of a daughter coming from a parent, and this is what this work demonstrates. We also ran it in one pot, so we could demonstrate that we both had the formation of ligated DNA strands and the formation of container molecules at the same time. But there was, I don't know why we couldn't, Cape, he was a very brilliant experimentalist, but we couldn't reproduce these results, so they were never published, so you'll have to be until next time we put it up. Anyways, so you can produce both of the building blocks by using the same type of chemistry. So now I'm going to switch to the problems, because that's where most of our, or all of our intellectual energy is put into now. For a while we've had this strategy that we want to anchor the DNA on the surface of this. Remember, it's on the surface of these vesicles, and we do that with these carbon chains, and then that is the way we think about doing the ligation-driven DNA amplification. We have a whole bunch of papers that are sort of related to this. So experimentally we have shown that you can anchor the DNA, and experimentally we have shown that we can actually also replicate the DNA when it's on the surface, but it's not with very, the yield is not very good. And we're somehow caught by what is called product inhibition. I don't know, you can imagine that if you have a long DNA strand and you have two pieces that are sort of aligning with it, so you have hybridization. So once these two pieces, they are glued together, you have a full plus and a minus strand, then it's much more difficult to get it away from each other, and you need to have these plus and minus strands to separate to actually have the process to go. Let me just get a little bit of water here. You need something to drink in the desert, right? Okay. So fortunately, again, there are people much smarter than us, and Julie's group up in Canada had came up with a much smarter way to do this reaction by what she called isothermal lesion-induced DNA amplification. The basic idea is that instead of having perfect matching plus and minus strands, you need to put in some errors so that you don't have such a good match. And then you also need to make sure that when they glue together, there's either a bulge or some other anomalies, a kink, so that once you have the plus and the minus strand, it actually likes to be separated. And she was able to show that you could easily get exponential, what's it called, amplification by doing that. And now we suddenly can use this method because, in the study I'm going to show you, we use this connection between thermodynamics and kinetics, where when we know the thermodynamics of, the free energy associated with hybridization, and we know the rate at which DNA molecules, they hybridize, it doesn't matter much about the sequence and the size, it's a very fast rate. Then it's just algebra from, I don't know, 6th grade or something. Then we can eliminate the little yellow one, which is the off rate, which is the critical one. So what we've done, we've then used Norman Packard's software that's AI-based design of experiment software, and then we have done simulations of the DNA replication, and then we've searched the space of different rates that will give us the fastest amplification or the fastest replication. So this, what you see over in these different colors, that is 100 experiments. These are simulation experiments. 100 experiments where the system, the AI system, proposes how we should change the parameters, and what you see downwards is how long time it takes to do the replication. And already in the second generation, the system discovers the best one, and it discovers a number of really good ones. And not only this system, it is actually able to tell us how the hierarchy of the different rates in this leader amplification scheme is rate limiting, and we were not surprised that it was the off rate, this product inhibition. But actually, if you tweak some of their other parameters, then it turns out that if you have a good enough off rate, then it becomes a ligation that's rate limiting. So it's nice to have this kind of tools. So these are the experimental results and a variety of results that this algorithm has found for us, and we of course have tested it and tried to understand why these are good. And so this is an exploration of how the amplification depends on the sequence composition, and it's not sort of the details of the sequence that's important. No, it's how many errors you have, how many what mismatches you have, and how you make either you put an A base in or bulge when you do the ligation. The other part of the catalyst here has to be able to transfer an electron. And this is done by an oxoguanine. It's a particular kind of guanine. In our first experiments, maybe I forgot to tell you that, in our first experiments, we simply just used the oxoguanine when it was not on a sequence. And then we demonstrated that you can use oxoguanine to control the process, because if oxoguanine is not present, then the metabolic process can't run. So now the next step is to put the oxoguanine into the strand. Here is an example where we have the two strands anchored to the surface. In this situation, there's different designs. We don't know which one is the best. I think part of it will have to be sort of experimentally determined, but we can do more calculations. I have a graduate student who wants to do that, so we can maybe do that before we go in the lab. But here we can actually calculate using some of the simple Marcos theory and figure out what the rate would be depending on how you compose these DNA strands and where you put the oxoguanine. You can put it up on the top, or you can put it down at the bottom. And you have the ruthenium complex, which is the one that can deliver the energy-rich electron. If it's too far away, it won't do much good. But you can actually put the ruthenium complex, you can have put an antenna on that so it can intercalate with the DNA directly, and then it's very efficient. So what we've done here, we've calculated what the charge transfer rates is, and it's maybe not so important with the details. Those of you who are interested, we can talk about that later. But we are convinced that we can actually that this will work. We are way better charge transfer than we need to, and if we want to slow down the charge transfer, we can do that by putting more mismatches in, and then compensate with more guanine and cytosine, because they are the ones that glue the more strongly the pieces together. So going back and looking at what actually happens when you both have a catalyst that's able to deliver an electron and it's able to replicate. There is a place where you have the sequence, so it just maintains the structure. If the sequence is either not good enough as a catalyst, as an electron donor, or as a replicator, you'll die. The system will die. But if you actually, let's assume that we put, this is again, we haven't done this experimentally, but let's assume you run the system out of the time. These are out of the x-axis here is how many generations you have of the protocells. And then if you allow the system to scavenge different types of oligomers, then it'll be able to assemble the ones that turns out to be a better replicator or a better catalyst. They'll of course make their system grow faster, and since there is individuality and inheritance, you'll actually have evolution. So some people like to say that this is right there at the onset of evolution. That is when life happens. I don't know. Maybe it's Anyways, you can. It's up to you what you think. But this is certainly a very important moment. Yeah, you can. Here is sort of a recipe for how you can measure this functional information, and I don't need to go into details. I think we will run out of time, but you can quantify that. So where does that leave us? So all the black stuff is stuff we have done in the lab. We need to have this DNA-controlled metabolism where we put the oxyguanine into the, we've gotten it to work with just the oxyguanine alone. We need to put it into a sequence. Our calculations say that this should indeed be possible. And also we need to use the leader method that Gibbs Davis group has developed so we can actually replicate this DNA. And I don't think they are insurmountable, and if we do that, then we have a fully functional protocell, where if we feed it with different oligomers, we'll be able to see evolution. So we'll be able to have a big party and maybe be the first ones who have actually created life, physical-chemical life, autonomous life in a test tube. But of course, when people sponsor us, they say, Steen, why should we give you money? Why is this useful? I mean, it's a funny thing because my physicists next door, that are looking for dark matter or doing other crazy things with high energy physics, they don't need to write stories about what dark matter can do for technology and how it's good for society. But, or the origins of the universe. But us, we have to tell the rest of the world has to talk about why it's good and why it's useful. I of course, do it because of curiosity. I want to understand what the creative forces in nature are. But I think there is a way out here. I think we can, once we have a self-replicator that can undergo simple evolution, we can feed it with we can decorate it with different functionalities, and then we can use similar experimental equipment that I talked about earlier to optimize it to actually do certain things. Yeah, I should say it costs money here. I think that we've probably gotten about $18 million so far, and I believe we can do it with 6 million, so we can do the rest of it because we've spent much more time preparing for the last part here. Anyways, this is our self-driving lab. We have not implemented that. You saw a simulation of it earlier. They could really speed up our work, and it could, of course, be used for automate these experiments, could speed up the process dramatically. Now, I just want to mention here at the end that evolution is not just evolution. There are two types of evolution we have identified. One of them is where you optimize, and the other one is where you invent genuinely new things. We call that evolutionary expansion, the last one, and the other one is obviously evolutionary optimization. It was until we really documented these things, a little bit of surprise when we looked at monocultures. This is Linsky's famous experiments where he has this E. coli that he's evolved, or he's sort of looked at for I think there's more than 100,000 generations now. And the interesting thing is that this system does not invent new fundamentally new properties. It's a monoculture, and it optimizes. If he changes the food source, it will optimize to this food source. So for you to actually have this guy to invent wheels or ears or whatever, the ability to fly, you need to provide this system with a more complex environment. And the reason why we started to study these things is because we know for physical system, this is magnetization. This goes the other way because it's, we have free energy. You go down the hill, and in fitness goes up the hill. But this is something we find everywhere. We can see it also in optimization, optimizing. This is photovoltaics. Over time, it really happens, follows the same optimization story. However, if you look at human technological and cultural evolution, there's something more going on, so you can then assume that it is optimization, and what is more is expansion. You can sort of say, now we subtract the optimization part, and then what's left is the expansion part. So to fully develop, or to develop these fully functional properties in a protocellular system, we have to feed the system with new things from the outside. And I know that some biologists get confused about this, but think about Margulis's hypothesis about how we went from prokaryotes to eukaryotes. It's not the prokaryotes that invented all these organelles in the cell. No, it simply happened by you aggregated functionalities. So this is actually a design principle I think we can use. I promise to say a few things about what I believe it means. Well, certainly from my perspective, it'll be really cool to understand how non-living materials can generate living materials, or how living materials can emerge from non-living materials, and also understand what the creative forces are in nature. Just by looking at one example, I think we could, usually that means a lot. If some of you may remember from history books that when the Wright brothers, they flew their first aeroplane, has a huge impact, even though it wasn't very useful, the first one. And ultimately, I think it will help us understand why we are here. Also, if you can make materials that have the same properties as life, then you suddenly can have these materials to disintegrate once you've used the technology. It has all the wonderful properties. It can evolve, it can learn, and it's adaptive, it can repair itself, and yada yada. But you can then have the materials be part of the ecosphere, so you don't, or the biospheres, you don't have all these issues as we do now, where we take resources up, we build technology, and we throw things out. We need to get into a sustainable situation where we can decompose our things. And of course, this is not just a new technology like this we have to invent. It's also, we need to invent new narratives. Unfortunately, we are still stuck in religion with the book of Genesis that says that we are something special, which is, of course, not right. I mean, it is true that we are different, but we are certainly part of the natural world. So I've been very fascinated by, of course I'm fascinated by the rest of you about what consciousness is. And I actually, when I arrived here, I read Thompson's article about sentience and life. And I think that since a protocell has differential responsiveness, I think we could view this as being a sentient being. So I believe, or the design principle I've used is this of dynamical hierarchies, that you by adding more and more materials to the system, new interactions occur, and you'll have observables that simply characterize life. And for instance, here, when you have a membrane, and then on this membrane you decorate it with the metabolic molecule, and you provide oil, then suddenly you have a metabolism. So you can observe a metabolism. You can't do that by at the earlier point. So could consciousness be sort of a human consciousness? Could that emerge in the similar way, just at a much, much higher level? That's something that, well, at least it makes sense to me. But then I want to say one word on here at the very end here about my confusion. And I know that we have different views on what consciousness is, and I think this is sort of the way I can contain my confusion. And I go back to Wheeler's circle of meaning. You have an observer that's conscious, that can make sense of the world, and the red circle somehow says that in the extreme situation says that the external world is experienced and constructed, and whereas the physical scientists, they tend to say, yeah, good, it's a good story, but look, you are made out of the physical world, and the physical scientists, they will then focus on the blue area here. But I think that maybe, and of course, it's not always peaceful, as we have seen here. People can really get into arguments about one or the other. But maybe the truth is this circular relationship. So that's what I will end up with here, and say thank you for your attention. And I think we will have questions.
All right, Dante, you come up. We have time for questions. Sara, are you there? Can we get Sara online?
Hello.
You're there. We hear. Can we see you? I can't.
We can't see you, but we can hear you.
That's probably good enough.
There we go.
Okay, good. Just leave it there. Okay, super. All right, we have a pretty good line of questioners, Sara, so please hang in there. And the first one, please.
First, I just want to say thank you so much for really wonderful talks that really go to the edge of these questions, especially on what life is. And so I just want to propose to all the speakers, especially to the first two speakers, we're talking about life as living organisms. What about the idea of life, like we're talking about consciousness, as a field from which consciousness emerges, or life as a universal principle, more in a field sense? And then organisms, once they become complex enough, can then express life through different systems. It's an idea we've been talking about for a long time, but I'm hearing it it's been abstracted or pushed away more, and I'm wanting to bring back this idea of life pre-existing that which is conscious, or consciousness even, as a universal phenomena.
Oh, Mike.
Either Sara or our other first speaker.
Sara, why don't you take it first? Dante's
Yeah,
waiting
sure.
for life.
Yeah, so I mean, I actually very much agree with that view. I don't really talk about it in terms of fields, but I do think even the things we call individuals are kind of instances of the underlying physics of life. So you might think of them as, like a particle is to a field or something. But so I can agree with that. The analogy I usually make is the kind of physics that I'm after is universal in the same sense that the laws of gravity are universal. But when you study gravity, you study, massive bodies or planetary motion or galaxies. And when you study life, you study the properties that we associate to individuals, or you study certain structures you see in the physical world. But they're manifestations of kind of a more general phenomena. So yes, I like that view.
Thank you.
Dante?
And I think that's partly why I'm here, to explore the different ways. What I showed mostly is the classic origin of life field, how the community looks at it very much as molecular building blocks, and if you could just stitch them together, then you would have all the answers. But I think Stuart's work was, kind of along the lines of what you're saying, right? Where you start to get into this consciousness preceding life and maybe being the driving force for self-organization. I'm very interested in that and to learn more.
Okay, yeah, just Sara was referring to it, but I'm thinking about life as pre-existing consciousness, or that consciousness emerges from life, but then systems or organisms emerge that are capable of transmitting these properties.
Okay, thank you.
That's an assumption that consciousness emerges from life.
Right.
Life could have emerged from... Dante's looking at... Steen, did you want to respond to her question? Okay. Next one, please.
Thank you for these talks. I love seeing the origin of life here at the Consciousness Conference. I think it's a great topic. And I actually think that this question of the origin of life, it kind of goes hand in hand with the hard problem of consciousness. And I suspect that they're ultimately one larger question that we're asking. So my question is, for all three of you, how important is water? And how much might we be overlooking the importance of water? I'm thinking especially of the unique physics and chemistry of water that might enable information storage and long-range quantum coherence.
Okay. I agree with you that I think maybe we're circling around the same thing with the origin of life and origin of consciousness question. That's what attracted me to this workshop and this venue and Stuart's work in the first place. How important is water? Right now, when we search for life, the strategy is to search for water, follow the water, look for water. We think it's essential, but it's viewed as the solvent where everything kind of happens inside of. So I think what you're asking is, could it be playing a role maybe that we're not considering? Right?
Yeah. Like, to what extent might we be looking at it as if it's too passive? And could it play a more active role in life and consciousness?
I don't have the answer to that. I think possibly. Yeah, Steen, maybe?
No, I think that water has, once you look at it, when you actually do this work in the lab or when you do the calculations, water is critical because without water you wouldn't have self-assembly. So we wouldn't be able to do any of the things that we are doing here without water, because water creates all these structures that enables, water creates structures around all these other molecules. So it is this interplay between these things that are totally critical. But there is, I wouldn't say that, I could imagine, but I don't know how, but I could imagine you could also have life in other solvents. For instance, we've actually done experiments where we have sort of gasoline-style liquids where we have droplets of water in there, and then we have it's called reverse micelles, where you have small containers of water, where we had the sort of the interesting metabolic process to happen inside these droplets that were immersed into this gasoline-style, this organic solvent. So. But it's just it costs a ton of money to do these things, and the funding agencies, they don't want to give us money to study life in esoteric materials in that way.
Sure. Sara, any comment?
Yeah, so I think the thing that's most interesting for me about thinking about the relationship between water as a liquid and life is that is to think about the liquid phase as actually being essential for open-ended evolution. And the best way I can explain that, which I think is related to your point about information processing, is to think about minerals as a more solid phase of life, because there's a lot of work done by Bob Hazen and different people looking at mineral evolution and the fact that a lot of the mineral diversity on our planet is actually generated by biological evolution. But minerals, because they're these crystalline forms, they actually they don't have the capacity for open-ended evolution in the same way that our wet chemistry does. As a sort of bedrock layer to support that. So I think there is something critical about having a liquid substrate for the kind of dynamics that leads to this cascading complexity that we've seen in our biosphere.
Yeah, and the other place where I think it's really important, it's a polar molecule, and when we look at the formation of the lipid layers or the boundaries that separate cells from the external environment, it's because of how they are polarized and how they are, one side of them is repelled by water and one side is attracted by water, that allows you to have that membrane in the first place. So in that sense, it is playing a critical role in one of the central components of cells.
It is the kind of the enemy of quantum coherence, though, but that's why you need non-polar. You mentioned quantum coherence. But Thank you. We better move on.
Thank you. Yeah, there's a long line.
Long line.
Thank you for the talks, and I'd like to present this question both to the panels, but also this question to the audience, as I'm looking to find others who are thinking in this way or asking these questions. So Avi Loeb reframed a quote about, if you want to make extraordinary claims, you need extraordinary evidence. And so in that theme, then, we need extraordinary inquiry methodology. So I'm curious, as Sara made the comment, is there a new physics arising? I would also ask, is there a new biology arising that invites us to think differently about how we think and measure, and maybe embody our inquiry differently? So I'm curious to know, what's being done within these organizations or in the audience about think tanks that are thinking about how we think and embody how we're inquiring. If we think about how we inquire as biology, as a humanoid in this space-time, will it give access to information of these fundamental questions?
Okay, I would say that's what I'm trying to do, right? In the sense that I'm in the position now where, I led this mission that claimed we're going to make progress on the origin of life. I'm trying to figure out if we can really deliver on that, and I'm not satisfied with the current state of the field and the current approach, the molecular approach, the assembly approach, to just trying to find the building blocks and finding a way to stitch them together, which is why we're talking to people like Sara, talking to people like Stuart, talking to people like Steen. So we're looking for that, and I'm hoping with the visibility of a project like OSIRIS-REx that maybe we can be a pathway to those kinds of new thoughts.
Quantum biology is sort of a new biology, too. Steen, did you want to
and I think that it's fair to say that both the community of consciousness and the community of the origin of life we're a little bit sort of children of science. We're not really accepted. It's not, you don't have departments at universities that do that, or they're sort of singularities if they are. So to have, so to say, organizational structures or platforms where we can actually unfold these things would be really useful. I mean, I can just tell you that we've been invited to CERN, because CERN, in the big collider accelerator community in Switzerland, because Stuart Kaufman was at a cocktail party with the director, and long story short, we were invited down, and the program manager was then asking us to come up with a way to so we together could go to the European community or commission to actually get sponsorship. So not only having CERN doing the origins of the universe and mass and energy and so forth, but also the origin of life. Turns out that the community at that time couldn't get along, the origins of life community. So we, but now there are several attempts, and I think eventually there will be centers for consciousness, I mean, at many universities, and there will be origins of life centers, but it's taking longer than I would wish it would take, certainly.
Sara, any comment?
Yeah, so I like the extraordinary claims require extraordinary evidence, but I like better that they require extraordinary explanations, because you can have the most mundane evidence for really deep ideas. If you just think about the evidence for motion, for example. So for me, it's more about how powerful is your explanation for the phenomena, and how deep does it dig into explaining a lot of the features of our reality. And when you look at it from that perspective, I think the thing that I find most interesting about the way we talk about the nature of life, and this is most apparent in sort of cultural dialogues about alien life, is that we're kind of in a mythological phase of development. In some sense, we have a lot of ways of talking about these problems culturally, but we haven't really been able to bridge sort of some of the deep intuition we have that is very historical in the fact that we're all living things, and we've been talking about what it is to be alive for centuries, into our like basic fundamental understanding in science. And I think to do that really requires a concerted dialogue between deep ideas in science and how we talk culturally about the nature of what we are and what we're looking for.
Thank you. We better move on, please. Thank you.
Hi, BJ Plaskin from Seattle. Appreciate these insights. Steen, in your thesis, I think you suggested that with the onset of evolution and thereafter, there's a gradual accumulation of information. But then prior to the evolution onset, there was also this baseline information that exists there. And what's more, it's a real, non-zero quantity that has the ability to self-maintain itself. So I'm curious if you might characterize what is that baseline information, and perhaps anyone else on the panel might want to offer some suggestions as well.
So I think that's a really good question, and in our system, then it is it's able to maintain itself. So it's this vesicle-based container structure where you have a metabolism, where it's just able to generate enough materials, and the catalyst is just able to replicate enough so that it can maintain. You'd be able to measure the yield over time, and it'll sort of fluctuate a little bit up and down. It's not that it will grow. For these things to grow, you need to have a more efficient catalyst. And this is just in our system, but I do believe, I completely agree with you that I think people have made sort of metabolically driven systems that can't evolve, but certainly can grow and divide and continue to do that. And that's why I like to think about life as being this gray zone, or whether things are living or not. You can talk about whether things are lifelike, and you can then add more and more properties until you've convinced even the most conservative that, yeah, this is indeed life. Some people would not agree that we haven't done it yet, but if we get it to work, that this is life. So there's also this story about definition involved in it.
Sara, any comment?
No, we can move on. That's great.
Next question, please.
Yeah, hi. My question is for Dr. Lauretta. And you had mentioned that the asteroids represent kind of the state of planets in the solar system very early on. So I was wondering if you're looking at this probe as sort of an archaeological investigation into what even Earth was like back then, or whether the hypothesis is that the building blocks of life had to be introduced externally to our planet?
Yes, great question. I think it is very much like an archaeological expedition. We're going back to the oldest rock layers, and they are the building blocks of planets. So since the asteroids have been preserved for 4.5 billion years, that is what accreted and grew up to build the planet Earth. The interesting thing about the asteroid like Bennu is that it has a lot of water, has a lot of carbon. That's very rare for asteroids in the inner main belt, and we think was even rarer in the inner solar system where the Earth accreted, because water, went from a vapor phase in the protoplanetary disk to a solid, we think, pretty far out, like around the orbit of Jupiter or so. So you had to have a way to bring that material condensed as a solid in the outer solar system into the inner solar system. So I like to say it was Bennu's siblings that were showing up from the outer solar system and landing on the Earth 4 billion years ago. So it does represent the kind of material that was available at the surface of the Earth, and from what the Earth itself was built from.
Thank you.
Sure.
Thanks. So I have another methodological question. This one for Sara specifically. I think it's a little bit more specific than the other one. Behind assembly theory, and some of it was actually sparked by a really awesome lecture that was titled on aliens, I guess, at, what was it, at the Santa Fe Institute a few weeks ago.
Oh, yeah.
Yeah, and I thought, by the way, I thought you framed the UFO thing actually really cleverly at that conference. But anyway, so a major focus of the origin of life, the field has been on producing molecules and amino acids and so on in labs and emulating and manipulating the conditions thought to be present in early Earth. And we saw this represented in some other presentations. But conversely, in thinking of the mess of the Petri dish of the early Earth's geochemical conditions, can you talk about the value of ensemble experiments where you you sort of embrace the many different kinds of chemistries and you alter the various geochemical conditions in hopes of finding self-assembly objects towards a living physics?
Yes, it's an excellent question. I think the biggest thing that's overlooked in the origin life field is how much of our agency goes into the design of experiments. So if you stop thinking about the sort of molecules of life as being decoupled from an evolutionary lineage, or anything decoupled from an evolutionary lineage, even the experiments we build in the lab are examples of life. And you're putting information into that physical system by virtue of you interacting with it. And so the sort of paradigm shift we need to have in origins of life is to move away from us being the designers designing the system, using all of the information of our evolutionary lineage, to removing as many constraints as possible, controlling for all the information put in on the boundary conditions, and then trying to see basically de novo how a chemical system can generate information on its own without us putting it there. And in order to be able to do that, you have to build automated experiments that can do batch reaction systems. And so this is technology actually that Lee Cronin's lab has developed. They have something called a computer, which is an automated system that can explore all of known reaction space and also discover new reactivity. And so I'm working a lot with his lab basically on how can we design experiments that don't include us as intelligent agents, or how do we control for the boundary conditions. And there's a postdoc at ASU Cole Math that's now working on how to quantify how much information has been put in past prebiotic chemistry experiments. You can basically say assembly in is what assembly out you get. The amount of constraints you put in is exactly what you get in the RNA you get out. You haven't generated any new complexity in most original life experiments. You put it all in by the boundary conditions.
I have a quick, possibly question generated by Sara. You talked about the vast complexity of the combinatorial space. Is that, in physics we have path integrals where you have an initial point and a final point and you explore all the spaces in between. Is that applicable here to that molecular construction method?
There is a feature of the spaces that you could talk about that, but it's not a physical feature of the universe, because the argument of assembly is the universe doesn't explore all possible paths. But when you get an object that's a high assembly object, all the pathways that are part of that object actually exist as an instantiation in that object. So in some sense, the further assembled you get, the more possible histories the universe could have had, but they're co-constrained by the existence of other objects. So not all of them are equally probable. They're completely constrained by that past history. So it's not exactly the same kind of principle, but it's in the space of ideas as being related.
Hi, I have two questions, one for Dante and one for Sara. Sara, actually it's a bit similar to what he asked, but in those causality chains and those space of possibilities, can we have feedbacks? Because if we can, so if we have feedbacks, then it reminds a bit of the IIT, Integrated Information Theory.
Yes,
Did you try to check the, like to calculate phi or something like that, to check for emergence in these spaces? I don't know.
Yeah, I mean, I've worked quite a bit on IIT. I think there's an interesting relationship between IIT and assembly in the sense that they both cut systems apart and then they try to look at the causal structure based on the structure of those parts. But there are some important differences because assembly theory is really unfolding systems in time, and IIT kind of, in some sense, it course screens time out, but you talk about like the transition probability matrix of labels of states. And so it's a different projection of what's happening in the system, and I'm really interested in the mapping between them, but I think there is a fundamental relationship. I just think they're looking at very different aspects of what these structures are. So that's the best answer I can give right now. I haven't actually done a formal mapping between them, but I'm also very interested in that question.
Yeah, thanks. For Dante, so you asked for new ideas for what else you do. But one thing I didn't hear you speak about the other theory of origin of life, not the RNA world, but the protocells world. What about that? Did you try to check, what was it?
The protocell world? You mean metabolism first model? We will be looking at those as well. We're looking for evidence of frozen chemical reaction networks. One of the big results from the survey of the asteroid that really surprised me was that we do see evidence that Bennu is a series of fragments of a much larger body that looks like it was shattered in the main asteroid belt maybe 800 to 1.4 billion years ago, 800 million to 1.4 billion years ago. And we see large deposits of carbonate veins, which show a hydrothermal system was active, maybe for millions of years over kilometer scales or even larger. So it looks like an environment very similar to the hydrothermal vents at the mid-ocean ridge, where we do think that there might have been, in the early origin of life on Earth, metabolic reaction networks, catalytic networks of the kind that Steen was discussing. I just didn't have time to go through everything that we're planning on doing right now on the project. I was even already over time with what I wanted to tackle today, focusing on some of the inspiration that I've gotten from Stuart's work mostly. But yeah, we do also consider the metabolism first model. We will be looking for evidence of chemical reaction networks in the samples to see if there might be some evidence of that. And particularly that it's an ancient hydrothermal system. That might be really interesting to compare to ancient and abiotic hydrothermal systems on the early Earth.
Thank you.
Okay, two more questioners, please.
Hi, thanks to all the speakers. My question relates to Dante and Sara's talk. So from my understanding of Sara's talk about molecular assembly is that from your work you found that only life produced complexity of like 15 or above. Is that correct?
That's right. Yeah.
Okay, so then I remember there was a slide in Dante's talk where he was showing this massive polyaromatic hydrocarbon. It seemed that would have a score above 15.
They're really highly symmetric molecules. So some of these large molecules that people talk about as being complex are actually not complex in terms of their causal structure. They're actually quite simple. If you have molecules with a lot of repeating units, then and you are recursively building them in the kind of structure of assembly space, they end up having very low assembly.
Okay, so that's not a sign of life.
I wouldn't argue a PAH is a sign of life.
Right, and I will say Sara and I have started the conversation about the molecular assembly index and applying that to the analysis of the return samples. I need to learn more about it. I have some questions still to come up to speed on how we might do that. The good news is we'll be doing lots of mass spectroscopy. That was one of the most interesting and intriguing things I found. In fact, when I first reached out to her, that was the first question. I was like, what can we do with the return sample in the context of your framework? And she's been thinking about that. I don't know if you want to comment on it, Sara, but there's a lot of targeted ideas to say if you're sending a probe to Mars or you're sending a probe to Europa and you're looking for signs of life and what you're getting back are mass spectra, she's got a really interesting approach that you could evaluate it. I do want to understand the score you gave the Murchison meteorite, what led to that, but that's what we'll talk about next time we get together, I think.
Yeah, we'll have more details. Yeah, but we are working on doing lower resolution mass spec so that you can actually use instruments on missions to detect assembly.
Hi, my name is Mike. I have a question for Sara. So I heard in the talk, great talk by the way, I appreciate it, and also in the talk you mentioned constructor theory, or how inspirations had came from it. I had briefly gone over a paper on constructor theory and the physical properties of knowledge. And I also have some separate interests, sort of parallel interest in information management, sort of building knowledge out of like cascades of that are similar to these causal chains that you have formed, and forming multiple of those. And in terms of like the knowledge for mine being these big chains of thought, I was wondering in terms of the inspiration you got for these from constructor theory on possibly the knowledge being these giant causal chains and the properties associated with them. Like for example, some you mentioned is like the chain length, or I think the chain length, I'm not too sure about the specifics, but I was wondering about what kind of properties you might have investigated, or if you're not investigating these properties of these chains, maybe where the inspirations from constructor theory sort of might have led you down certain paths.
Yeah, no, I'm happy to talk about it. I have worked with Chiara Marletto for a number of years, and I've interacted with David a bit. But Chiara and I have yet to write a paper on all the ideas we've discussed over the years, but one of them was specifically on the idea of the like an arrow of knowledge, that the biosphere is basically increasing the knowledge. But the problem at that time, this was kind of pre-assembly era for me, was how you actually quantify that. And I think that is actually a feature of the assembly spaces because of this causal chain structure, which it sounds like is very similar to the kind of things you're thinking about with quantifying knowledge. So I think there's definitely a formal way of doing it. The other thing that I really love that's a constructor theoretic concept is I think that they have the closest concept to what information actually is, in terms of thinking about information as things like a property that you can copy between physical media and it retains its causal properties. And so they have a lot of ways of talking about information that I think are much more physical than in any other way I've ever read about the concept of information in any literature. And I think if you think about that property, there's actually ways of thinking about those properties they identify in assembly spaces that I think are really interesting. And so that's actually where a lot of the sort of constructor theoretic type thoughts I have right now are.
Very cool. Thank you very much.
All right, well, thank you, everybody. Sara, Steen, Dante. Let's give them all a big round of applause for a great session.